All right, so now we're going to go and we're going to meet the operon. So we're moving on into gene regulation. So what controls our genes? Because we don't need every gene transcribed and translated all the time, right? That would be inefficient and a waste of energy. So I'm going to switch over to my PowerPoint view and see if I can do this, because where's my play? I just like sometimes when you do them on the PowerPoint instead of the Google Slides. So this is new information. Oh no, I had video panel, but that was me. So you can have housekeeping genes. These are just like genes that you need for everyday life, right? Essential genes for living cells, like genes that would code for mitochondria's cytochrome C, for example. That's essential for making energy in respiration. They're expressed all the time. Then you also have inducible genes. So genes that are off to begin with, and then you can induce them to turn them on. So they're only going to be expressed when needed. And then you also have repressible genes, and these are going to be turned off when needed. So these are typically on all the time, and then off when you no longer need them. So bacteria need to respond to their environment quite quickly, right? Because they're a very little organism, and so they're going to be affected by a lot, or affected greatly by their environment. If they have enough of a product, they don't need to make any more of it. So they're going to stop the production. So these repressible operons are usually going to be like when you're synthesizing something. So they stop the production of the enzyme so that you no longer make whatever the product is. If they find a new energy source, they need to utilize it quickly, and so they need to be able to break that down. So they need, of course, nutrition for metabolism, growth, reproduction. So they're going to start production of enzymes for digestion. So we're going to stop when we no longer need something. We're going to start when something becomes available, basically, right? We don't need to build any more of this because we have enough. This, oh, something's there that I didn't have before and I need to break it down. Okay, so we're going back to our allosteric regulation. Remember, you had your enzyme, you have your active site, and the allosteric site. So that allosteric site is what we're going to make use of in this case. The allosteric site, if we bind to it, we might change the shape to be usable, or we might change the shape to be not usable. So inducible becomes usable, repressible, not usable. So the product, this is that negative feedback loop, and we've used this with tryptophan before. So in this synthesis reaction, we're building tryptophan. When we have more than enough, tryptophan comes back, acts as a regulator, binds allosterically to this enzyme, and stops production of the rest of the process. So instead of blocking an enzyme's function, we can regulate the genes at any point along the way. So this is going to save energy and resources along the way. So different cells, obviously, like our skin cells are different than our eye cells, right? And what makes them different? They're expressing different genes. So not all genes are being expressed all the time. And this is true for the bacteria as well. So we're going to start with the prokaryotes today, and then we'll get into eukaryotes, which is a little more complicated later. So, so we can regulate gene expression by turning genes on or turning genes off. So turning them on is inducible, turning them off is repressible. So when they have enough tryptophan, they don't need to make any more, so they're going to turn that gene off. Another example, this is a turning on example, and quite often we use the Lac Operon. When lactose becomes available, which is a type of sugar, then the Lac Operon is induced to create the enzymes to break down the Lac Operon, or the lactose, sorry. So repressible, inducible. So here's the operon, and you absolutely need to know the components of the operon. I'm giving you two examples, the Trip Operon and the Lac Operon, which the AP uses over and over again as illustrative examples. But know that if they gave you any other molecule, the operon structure is the same. They all are going to have a regulatory gene upstream right here that codes for the regulatory protein. They're all going to have a promoter sequence. We've talked about the TATA box. They're all going to have a promoter sequence where the RNA polymerase will bind. They all are going to have these structural genes. Each of these genes codes for an enzyme, for example, along the process. They all have an operator right here. This is what is going to turn the gene on or off. This is like the on-off switch. The regulatory gene here, the regulatory protein will bond to the operator. And that either turns it on or turns it off, depending on if it's a repressible or an inducible process. This is confusing to say the first time, and when I'm talking, you're just like, oh my God, there's all these words coming in and I don't know what to do with them. This isn't a one and done topic. We will spend a few days going over this and trying to make sense of it. Also, on your own, you should draw these out to try to make sense of it as well. So the operon genes group together. So this whole piece is called an operon, right? So you have the promoter right here. This is where RNA polymerase will bind. See this structure here? This is the polymerase. So this whole section is your promoter. So if the operator is open, the polymerase can go through and transcribe. If the operator is closed, if this is bound to it and blocking it, then the polymerase can't move and it won't transcribe. So that is how they're going to control it, using the operator and the repressor protein. So all operons have an operator, a promoter, and their structural genes. So this is your mRNA, these are your genes. Here's your repressor protein bound to the operator. I need to change my font size. Polymerase upstream. So we have our inducible operon. The operon is typically off, turned on when needed. So this is regulated by a substrate. So something present will bond to the repressor, which then will bond to the promoter or the operator, which turns it off or on. I'm sorry, I'm talking about the other one right now. The inducible, the repressor is typically bound already to the regulator, then the substrate binds to the repressor, pulls it off. Then you can proceed with the process. Sorry about that. So it's off when not needed. The repressor protein is the one I was just accidentally saying because I thought that one was first. So the operator is turned on and then off when no longer needed. And our two key examples are the Lac Operon and the Trip Operon. So this is tryptophan. So here you see polymerase is bound. The repressor is sitting in position. So in this case, it could not go anywhere. My eyes, this is just a bunch of, these used to come in one at a time. That's why I had it in my PowerPoint, because it doesn't do it in, you don't care, I'm sorry. So the synthesis model, when access to tryptophan is present, it binds to the trip repressor protein and triggers the repressor to bind to the DNA, which blocks the transcription. So your repressor protein here, when tryptophan is present, it'll bind allosterically. Then the repressor will bind to the operator, and then that stops transcription because now it's in the way. RNA polymerase can't go anywhere. So if tryptophan is present, we clearly don't need to make it. It's already there. So we're going to turn it off. So this is showing you The regulatory gene codes for the repressor protein. Tryptophan binds to the repressor protein. Notice the change in shape, conformational change. So this shape would not fit the operator, but this shape does. So when you have that conformational change, it changes its shape to fit and binds to the operator. Polymerase can't go anywhere, so you don't transcribe anything. I'm going to skip past these because I just think it's easier to look at those slides. The digestive model is when something's present that is new to your environment. So typically, in this case, the regulatory protein, the repressor protein, would already be bound to the operator. When its allosteric regulator is present, in this case, lactose, so lactose would bind to this promoter, or I'm sorry, this regulatory protein that was sitting here at the operator. Notice it changes shape to a shape that no longer fits the operator. So this causes the repressor protein to be released. Now, polymerase is not blocked, and it can carry on its process of transcription. So it all has to do with whether that repressor protein is bound to the operator or if it's releasing the operator. So repressible proteins are typically in anabolic pathways, those that are synthesizing products. The inducible operator is usually in digestive or catabolic pathways. We need to break down something that's present. Can you identify what each of these structures are? What's this purple box? You guys can shout out loud, or you can, so yes, well, this purple box is not the repressor. It does code for the repressor, but see, it's part of the DNA sequence. So this is the regulatory gene that codes for the repressor, yeah. And what would this teardrop shaped thing be? That's the polymerase. And the yellow, what is this yellow Lego looking thing? The whole thing is the operon. That's the operator. Good. And then you would refer to each of these as what kind of genes? They start with an S. Structural genes. Yeah. Okay. So for this trip operon to be on, is the promoter or is the repressor protein bound, or is it released? First of all, for it to be on, for it to be turned on, so transcription occurs. Is the repressor bound or is it released? I mean, yeah, it's released, yeah. So it's already going to be off, which means tryptophan levels must be low. If tryptophan levels were high, it would bind and it would stop the process. So this is our Lac Operon. Same thing, regulatory gene, mRNA, repressor, polymerase, operator. So if this repressor is bound, is lactose present or not present? In this particular case, it must not be present, right? Because lactose would bind to it and release it. So in the first one, in the repressor, the product binds and turns it off. And then in the second one, the inducible, the repressor is released and it's turned on. Okay, that can be difficult to talk about. So we have, I think, 10 minutes. So you guys are going to do this. There's a video, a Bozeman video. It's 10 minutes. Read. You can highlight. You can change the color of the font for things that you find important, but I want to see some sort of talking to the text, whether that's colored font, circling, or highlighting, and then you're just going to label and summarize just the lactose operon when lactose is present or absent. Okay, so what questions do you have?